Well, I'm Dr. Jim Del Rosso, a dermatologist in Las Vegas, Nevada, and proud to say that I'm the editor-in-chief of the Journal of Clinical and Aesthetic Dermatology. And I'm thrilled today to have another JCAD TV. And I couldn't think of a better person to have for the particular topic we're going to be talking about today is my good friend, colleague, and also the president of the Nevada Society of Dermatology and Dermatologic Surgery. And I'm happy to say I help him with that organization, is David Cotter, who has quickly become a legend, not only in the Las Vegas community, but across dermatology. Great to have you here today, David.
Thanks, Jim. It's always a pleasure to chat with you.
So, we're going to be talking today about what I think is one of the biggest advances in dermatology that we've had in a long time. And typically, that's talking about a treatment. But this is a diagnostic test that allows us to select treatment better. And it's the advanced AD-Tx. And when I look back at what was done a few years ago in 2023 in an article that was in the Blue Journal, came out of both Yale and Stony Brook, they looked at baseline characteristics by RNA hybridization testing to look at different cytokine profiles in atopic dermatitis and look at responders to dupilumab. And they came up with three major groups, though there's certainly gradations in between. They came up with a group that is a very high level of Th2 pathway involvement. And that's primarily IL-4, IL-13, and other things like IL-31 kick in. And those patients were typically very, very high responders to dupilumab, which inhibits IL-4 and IL-13. But then they found two other groups. They found a partially responsive group that had Th2 involved, but also started to integrate some other cytokine pathways like IL-17, IL-22, and some more Th1 activation. So they were partial responders. And we started to see that in clinical practice where they were a lot better than where they started, but they didn't respond as well as that other group. And then there were the poor responders where their cytokine profile had less of that Th2 dominance and was more interferon gamma. So now we're at the point where we have a personalized test, which helps us break down patients into two different groups. And I'm going to ask you in a moment, David, to explain that test. But basically, the way it turned out was there is a Th2 molecular profile that's identified that's maybe three-fourths of the patients, so 7 out of 10 patients, and that will tell you that they're going to respond to dupilumab or an agent that inhibits IL-4 or even IL-13, but will also respond equally as well. They respond the same to a Janus kinase inhibitor. But David, I know you talk about this a lot. We have that 1 out of 4 that doesn't do so well on one of those monoclonal antibodies and falls into a pattern where Janus kinase inhibitors work a lot better. So we now have a test that we can use up front to identify these patients so we don't have to guess. So I'm going to turn it over to you because I know you know a lot about this test.
Thanks, Jim. And you're absolutely right. That's a great historical overview. Certainly, with atopic dermatitis, there's myriad drivers of disease. We know about IL-4 and type 2 inflammation and Th2 cytokines, like you very elegantly laid out. But certainly, that's not the whole story. There are some patients that have more broad immune dysregulation, and because of that, they need more comprehensive treatments. But we can't predict which patients need which drugs just by looking at them. The clinical phenotype doesn't always match up to the therapy that we think is best. And the trouble in picking one of our 6 FDA-approved systemic medicines is not necessarily knowing which drug is going to work best for any individual patient. Certainly, we know that if you just put all comers on a JAK inhibitor, odds are that they'll do better. We have head-to-head trials that look at upadacitinib versus dupilumab, abrocitinib versus dupilumab. We have network meta-analyses that compare the JAK inhibitors against the biologic. And over again, whether it's a clinical trial or an NMA, JAK inhibitors edge out the biologics on average. But that doesn't mean every patient needs a JAK inhibitor.
But David, isn't that really what's separating out these patients that don't have that high as much Th2 involvement. That's what's showing us that difference, and this allows us to predict who they are.
Well, let's get into that. Let's talk about the Castle AdvanceAD-Tx test. Let's talk about the identity clinical trial, which is what led to the development and validation of this clinical tool, which is a gene expression profiling test to help guide which patients truly need a JAK inhibitor or which ones could go on a JAK or a conventional biologic. To answer that question, Jim, what identity was a multi-center study that looked to create a gene expression profile that could predict responses to therapy. There were hundreds of patients in the initial phase of doing this development, but in the independent validation study, there were 110 patients with moderate to severe atopic dermatitis that had gene expression profiles that map to 12 different pathways, things that are super important for cytokine signaling, Th17 differentiation, chemokines, initial T cell activation, keratinization, and many other aspects of T cell biology and skin biology as it relates to atopic dermatitis. Each one of these key pathways, one of these 12 pathways, has the gene expression profile applied to an independent neural network that gives you a gene expression profile score of 0 to 1 for each one of these pathways. And then you get a composite score between 0 and 12 that tells you if your patient is more likely to be a Th2 molecular profile or a JAK inhibitor profile. And what that actually means is response to therapy. And to your point about these JAK inhibitor responder profiles, these are the most important patients to call out. In the identity trial, if you look across the board at all comers, only 25% of patients hit an EZ90 by 3 months. When you look in the Th2 profile patients, it doesn't matter if they were on a biologic or a JAK inhibitor. The rates of EZ90 achievement were very, very similar. They were both cruising right around 25 to 30%.
But that was the ones that were Th2 profile, correct?
Yes, exactly. The Th2 patients, which, like you said, about 70% of patients in the study had a Th2 profile, didn't matter if you put them on a biologic or JAK inhibitor. They responded equivalently to either medication, whether we're talking about EZ90, itch response, flare data, or DLQI. But the big takeaway, the big exciting punchline is if your patient is a JAK profile responder and you put them on a JAK inhibitor, they beat the spread, Jim. Instead of running with the pack, the average EZ90 by 3 months happened in 25% of people. It was 45.5% of the patients when they have a JAK profile that go on a JAK inhibitor achieve an EZ90. And that's not even the most striking finding from the study. What to me matters the most is what happens when you take a JAK profile patient and put them on a biologic. Only 8.3% of those patients achieve an EZ90 by 3 months. And to me, that says, well, one, if you've got someone sitting in your clinic that truly needs a JAK inhibitor and you don't offer them one, they're not going to get to where they need. Only 8.3% of patients with a JAK profile that get a biologic achieve an EZ90 by 3 months.
So what that means to me is that individuals that are averse to the idea of using JAKs for whatever reason they're not using them. If they were, I'm going to roll the odds here in Vegas, lucky enough to have a patient that was predominant Th2 and they used the conventional biologic therapy, let's say dupilumab, they would hit that jackpot because that's the way it turned out. But if they have that 1 out of 4 that did not, that patient's not going to get the opportunity to have that optimal benefit. And my understanding also, David, is that it was 4 times faster to even get to whether it was clear or almost clear or EZ90 when they used the JAK in the patient that had that JAK responder profile, correct?
You're absolutely right. Not only do JAK patients hit their EZ90 more consistently when they get a JAK versus biologic patients, they hit it 4 times faster. To me, the reason why this matters so much is if you're sitting there and you're seeing a patient and they're not responding to their biologic the way that you would have expected or they would have hoped, and your next step in therapy, it needs to be to use the AdvanceAD-Tx test before you just switch them to another biologic. You need to figure out if this is a patient that truly needs a JAK inhibitor. But better yet, use the AdvanceAD-Tx at baseline. We're using it to guide initial systemic therapy choice because even though, Jim, you and I both live and practice in Las Vegas, and historically we have to play the odds, I don't want to gamble on my patient's care. If I can do a test that's going to guide the decision of which therapy they're going to do better on, we're doing that in all of our patients to guide initial systemic therapy choice now.
So that's why couple of times when I was presenting this at meetings. To me, this is one of the biggest advances that we have because we did have a test from a different company with psoriasis, and it turned out there wasn't as much of an issue to really pick an agent that was helpful in psoriasis. We didn't have as much of that differentiation, but it's very different with atopic dermatitis, not only from the clinical appearance, but the symptomatology and the effects of this disease. So that's why I think the DLQI, quality of life data, is so important in that particular trial. And if you can tell that up front, it's an easy test, right? A little bit of a scraping, it's not invasive. You send it to the lab, and I'd like you to talk a little bit about that process, how easy it is, and how and the accessibility of it, because I know you're actively using it and have studied it. But it really allows us up front to figure out how to give us the best chance of getting it right in these patients.
Yeah, you're absolutely right. The test is so easy to do in clinic. If you've ever done a KOH prep, you can do this test. If you've never done a KOH prep, that's okay. I'll walk you through how to do it. You find the patient's most severe area of eczema, or maybe the area you are hoping to impact to the greatest degree utilizing your therapy. And you clean the area off with a little alcohol wipe. I always warn the patients sometimes the alcohol wipe can sting on inflamed skin. And then in the kit that Castle sends you, are two curettes. You pull them out. There's two little test tubes that have an RNA buffer. You pull them out, and I gently scrape with the dull side of the curette about 10 times on the most severely affected areas. I inspect my curette to make sure I have some dead keratinocytes. You're not causing bleeding. You're not going deep. This isn't an EDC, ED&C. This is just a gentle curettage of the hyperkeratosis, the epidermis, the scale, and then you drop it into your test tube, swirl it around, and make sure that those skin cells fall off. Put the lid on and repeat that a second time 2 centimeters away. So you do two spots 2 centimeters apart. The reason why is in case for some reason there wasn't enough genomic material to generate RNA from the first sample, you have the second sample as a backup. And doing two samples this way is a very high fidelity rate from the company. You have to ship it out within 3 days of collecting it, and you typically get a result back within 5 to 10 business days.
What about coverage for the test?
Right now, the coverage is somewhat undetermined. Castle's running the test with a plan to run the coverage the same way they do their melanoma test and all of their other gene expression profile tests, which means that certainly they'll bill the patient's insurance, but in the entire history of the company, they've never balance billed a patient, which is great because it means I can do this test without having to worry about running up the cost of care for my patient or breaking the bank, and I can get them to their ultimate therapy sooner. It's so important to pick the right systemic at the first step because when you put the patient on the wrong drug, not only do you waste time in their life, but you also increase the cost of care by cycling them through a medicine that's not going to be effective.
What I like about it is knowing up front, because we had typically always thought that if a patient didn't respond to, let's say, dupilumab, that we now have some data with some of the other agents, the anti-IL-13s, that we can go to those. But if we know that the patient doesn't fall into that Th2 profile, we would jump over that step because it's not likely going to work. And we know that based on this test, right? So it provides us with so much more ability to see what is likely to happen than what we had before. That's what's so exciting to me.
I agree with you. The idea is we can break biologic cycling, we can overcome therapeutic inertia in the right patients. Certainly, there's patients that absolutely need JAK inhibitors. We see that from the JAK inhibitor clinical trials where they are superior to at least dupilumab in head-to-head studies. But it's not necessarily that 100% of patients absolutely have to be on a JAK inhibitor, but the ones that need it, they really, really do. And probably we have a subset of patients that are super responders to JAK inhibitors. You've got your average patients that do well, whether they're on a biologic or a JAK. That aligns nicely with the Th2 molecular profile in the identity study. But then you have these super responders to JAK inhibitors, and those are your JAK profile patients. And the flip side of that coin is if you're a JAK responder, you're a biologic underperformer, which is a very important point to drive home. A lot of patients who will start on a biologic, if they don't have a good result, they might not ever follow up. Sometimes people discontinue therapy and they're lost to follow up. In fact, about 40% of conventional biologics are discontinued, generally due to treatment ineffectiveness. And the question is, what happens to those 41% of patients? Some will cycle to another therapy, and some just won't follow up.
Some of them have been around the block so many times that they just give up. But I have a question about that middle zone. So you have patients that were severely affected by atopic dermatitis. They were miserable. Significant 50% involvement on the skin, difficulty sleeping, itching. That's their baseline. And now they have a good response to the biologic, but not the optimal response. Those people I'm also concerned about because they'll come in and say, "Oh, Dr. Cotter, it's nice to see you. Hey, how you doing? Oh, I'm doing pretty good. I'm okay." You never really find out that they're not doing that well, or that maybe they're not sleeping as well as they were, or they're itching more because it doesn't come up in the conversation. But there's that opportunity to miss the fact that they could have a better response. And this test would tell us that by simply saying this patient is a Jack responder, right?
100%. And Jim, I encountered that just about 2 weeks ago. I had a new patient come in. He'd been on a biologic for a number of years, but was still requiring high topical corticosteroid usage, but overall was generally well controlled. Although he's still at 3% BSA, he was still 4 to 6 out of 10 on an itch scale, depending on the day, and he had moderate disease. He was better, but not better. He'd improved, but he'd not cleared. And we scraped him with the Castle test, and he came back as a JAK inhibitor responder. So drives home that point of exactly what you're describing. These are real world scenarios where patients are improved, but their therapies have stalled out. They've essentially plateaued. And now to get them over that hump, having additional information to know if we should stay the course with a biologic, switch to a different biologic agent, or change classes entirely to a JAK inhibitor. This is so helpful to break the cycle of just randomly rotating therapies based on, how we're feeling that day or what paper we read the most recently. We can have objective data to bring precision medicine to inflammatory skin diseases.
Absolutely. I mean, about 4 years ago, I actually had written an article for a textbook on atopic dermatitis for Nick Brownstone, and he asked me to look at the future and also to look at personalized medicine. And back then, I was just so excited about the possibility that this can happen. And here we are, 3 or 4 years later, it's actually happening. I mean, normally things take decades to go from one step to another. So I am really excited about this test. Not just because you get me excited to talk about anything in dermatology, David, but it's the real deal. It's why not?
To me, this test is going to help our patients get to their ultimate treatment faster, which is ultimately a great
Right.
thing. We want people hitting their DLQIs of 0 or 1 as fast as possible. So many of our patients, Jim, as you know, have been living with eczema for many years, and that's too long. And from the first visit with me, I want to make sure I'm picking the most effective treatment for them. And now having some additional objective data to guide that decision is really nice.
Oh, Dr. Cotter, does it hurt? Absolutely not. You're not going to feel it. Does it hurt me financially? No. Right? So it's a win-win.
In my mind, there's not been a reason to not use this test on any patient who's 12 or older to guide initial systemic therapy selection or subsequent systemic therapy. The most common areas we're using it are new starts or switches. Switches because of low efficacy or because of an AE. Maybe someone's doing great on their biologic, but they get conjunctivitis, they had some joint pain, or they got a red face. There was something that they didn't like about it. Maybe they had an injection site reaction, but they did great. Do the scraping and make sure they really do have a Th2 molecular profile before you switch them to another biologic.
David, thank you so much. I'll ask you one more time. Do you have any parting words on this before we sign off on the ultimate program, JCAD TV, right?
The last thing I'll say is that this test is so easy to do. It takes less than 30 seconds. And those 30 seconds extra you spend with your patient might mean getting them to their therapeutic goals 3 to 4 months faster, maybe even a year faster than just cycling through whatever the next biologic is that you're choosing.
Absolutely. Thanks a lot, David. I appreciate your time as always.
Thanks, Jim.